Thank you very much to the organisers for inviting me to present at the COVID-19 Symposium. I'm going to be talking about testing for the SARS coronavirus 2 in the laboratory and also in the community. So the outline of my talk is that I will talk about a bit about the virology and then remind ourselves of how the diagnostic tests were developed, the ways in which the different ones have been used and the reasons behind the thinking behind that, and then some evaluation of the three tests: the polymerase chain reaction, serological test, and also the lateral flow devices, which I'm sure you're all very familiar with. And then we'll start to have a little bit to think about what we've learned from all of that, and then also looking at where we are with testing now in 2023. So at the time of developing this presentation, which was March 2023, there were over 700 million confirmed cases since the first case was recorded at the end of 2019, and almost 7 million recorded deaths. And at the time when you are accessing this presentation, there'll probably be a little bit more than that as well. And the other thing to note is that there have been 13,000 million doses of vaccine. And that's quite an important thing to remember when people are worrying about the safety and efficacy of the vaccine, that we have an awful lot of data about that now, because there have been an awful lot of doses with people all over the world and all demographic groups. So that we have a lot of information about how effective the vaccines actually are. So a little bit of virology. It's a positive sense single-stranded RNA virus. And coronaviruses have a very, very large genome and unusually have one continuous strand. They're enveloped, which is one of those things that virologists used to have to learn. Part of their special exam was whether something was enveloped or not. And the reason is because in order for it to be a viable particle, it has to have that lipid envelope around it. And that means it can be killed by detergent. So it's actually a very useful thing to do in terms of infection control. But within the envelope of coronaviruses, some of the key glycoproteins, some of them you've probably heard of, are actually embedded in the envelope. So they're produced by the virus particle, but they get embedded in the envelope at the time when the whole virus particle gets assembled. A little bit of classification, and we'll see here that the family of Coronaviridae and the subfamily of Coronaviridae, the genus is beta coronavirus, and the species is severe acute respiratory syndrome related coronavirus. And within that whole species, there are lots of different viruses, including SARS-1, Middle Eastern respiratory syndrome coronavirus, SARS-CoV-2, but also a number of VAT SARS coronaviruses, and also the civet SARS coronavirus, which we think was the intermediate host for the virus, which turned into SARS-CoV-1, the 2002-3 outbreak. This is a picture of what coronaviruses look like, and this is taken from an old textbook of mine. So this is the common cold coronavirus. But what you can see is this is the spike glycoprotein, the thing that you've all heard of, because that was the basis of the vaccine and also for some of the virus testing. And that's kind of embedded into the lipid envelope, and you'll see that it's quite a diffuse and uneven shape. People often say that it's called coronavirus because it looks like a crown, but actually as you see here that I've underlined here, it's actually named for the solar corona, where the sun goes behind the clouds, and then you get that diffuse rays. So although obviously it's the same Latin root, it isn't named for a crown. The key antigens in all coronaviruses that we're looking at would be the spike protein, which is sticking out on the very edge of the envelope. There's also the membrane glycoprotein, which is embedded within the envelope. And then there's a non-glycosylated protein, which is the envelope protein, which again is embedded even more deeply into the envelope. And then there's the nucleocapsid protein, which is forms the outside of the internal sort of viral particle. In terms of immunology and diagnostics, it's considered that the spike protein and the membrane protein are considered to be the most immunogenic. Although in terms of diagnostics, the nucleocapsid protein has also been quite important. This is a very stylized diagram of the SARS-CoV-2 genome. And what you can see here, what I want you to kind of notice here, are that at one end, you have the open reading frames, which are the coding for non-structural proteins, including the RNA-dependent RNA polymerase, which is a key part of the replication cycle for coronaviruses. And then on the other side, these are all the structural proteins. So there's the code for spike, envelope, membrane, and nucleocapsid. And just for note that this is going to be worth noting for later on, is that there are 1,273 amino acid residues in the spike protein. The spike protein is a key element because it binds to receptors on human cells, the angiotensin converting enzyme ACE2, and that is a key target, so therefore it's linked to pathogenicity, and that was why it was one of the reasons why it was made the target for the vaccines. And SARS-CoV-1 also has that same receptor target. It's just worth noting that the Middle Eastern respiratory syndrome coronavirus, which is very closely related, as we noted earlier, binds to a different receptor, the so-called dipeptidyl peptidase 4, DPP4. So that it's a slight difference, but it's still the principle of the fact that it binds to a particular cell surface protein on human cells is the same. So in terms of testing, we have, generally in virology, we have two methods for testing these days, and that would be the polymerase chain reaction or some other kind of nucleic acid test, and then serology. And those are the ones which have actually were implemented for testing for SARS-CoV-2 in clinical laboratories. But then community testing, there was mass PCR in labs outside of the NHS in England, and similarly outside of the normal hospital laboratories in other countries, and also the rapid antigen test, which I'm sure you're all very familiar with now. It's just worth noting, because we're going to talk about the way the tests developed and were the ones which came to be the most useful, the ones which where people understood this is the timeline that you need to notice for virus detection. So the incubation time was considered to be 10 to 14 days. Sometimes it was a little bit longer, but and as the virus changed and there were the new mutations came along, some of the new variants, the incubation time was on average a little bit shorter than that, but certainly up sort of 10 to 12 days. And then at that point, that's when the patient becomes symptomatic. And what you'll note from here is that there was a time of about a day or two when, with very sensitive tests, the virus would be detectable in the respiratory samples just immediately before the patient got symptomatic. This is not unusual for virus infections, particularly respiratory virus infections. But it takes at least a week after the symptoms develop for even IgM to become detectable in serum. And obviously that should be detectable for quite some time afterwards, 2 or 3 months, but it's at least a week before it becomes detectable after the symptoms have developed. So for the polymerase chain reaction, there were a lot of different tests which developed. Most of the kits that became available and now are now available use 2 or 3 sets of primers, one site on the open reading frame end of the genome, and then the other one at the structural protein sequence. Quite often it was the M or sometimes N, sometimes envelope, and sometimes spike as well, depending on the different kit by the different commercial companies. So once the sequence had been released by the Chinese scientists to the World Health Organization, the people started working really hard to develop the tests. And what I think is really remarkable is that how quickly the tests developed by the across Europe, people through the ECDC, actually, people all got together, including people from the influenza reference laboratory here in the UK, and got together and worked to develop a protocol which became available by mid-January of 2020. We had a protocol which was rolled out for reference laboratories, and then commercial kits became available in towards March and April of 2020. This is just an example of what a PCR output would look like, and what we've noticed. This is a kit that was being. This is a commercial kit that was used in a lab that I was working with, and this is the notice that the green line is the open reading frame primers, and the red line is an envelope gene. And in fact, that was just a general envelope protein for all coronaviruses, whereas the green one is a specific for SARS-CoV-2. And the blue line here is the internal control. So what this tells you is that the PCR protocol has actually worked, but no virus has been detected. This is a one where the genome has been detected. You see again, here's the internal control, and then this is the open reading frame this is the open reading frame 1A and B for the COVID, for SARS-CoV-2, and this is the general envelope gene for which was common to all coronaviruses, which is the second thing. So what that shows you is that both of those bits of gene were being detected. And the polymerase chain reaction, of course, is considered to be the gold standard, has excellent sensitivity and specificity. At some point in the whole sort of COVID the cycle, the last 2 or 3 years, where people have been discussing about what's the best way to deal with it as a sort of in terms of public health in general, there were people did start questioning whether it was too sensitive because it's detecting the genome. Is it always picking up live infectious virus? This isn't something that we would really have talked about in the general public outside of the pathology departments up until that point, and it's not something that we would necessarily think about in terms of questioning the results of any other PCR reaction for any other virus, because you set the cutoff at the right point so that you know that this is definitely a live infection. However, I'm mentioning it because it was people were questioning it. The really good advantage, and we found this out as time went on, the use of at least 2 primers was really an important thing to have done, because what it allowed to happen was that as there were new variants arose, in some cases the variant wasn't picked up, that the change in the genome meant that the primers didn't work on picking up one part of the genome, but they did pick up the other part, or in case there were 3 primers, 2 of them it did pick up and the other one it didn't. So people in laboratories were starting to report that there was weird results. Was it positive or was it not? And it turned out on investigation it was because the variant was giving an anomalous result in the PCR test, but because of the number of primers, it actually allowed for that to be picked up really early. So that was a good thing about PCR, the way they were set up. Just reminding you of the genome again. So again, reminding you of the 1273 amino acid residues. And the reason for that is because over time, you probably won't be too surprised, I've been tracking the mutations and looking for what the key mutations are in the different so what the key changes in the genome were in the interesting mutations. So I've called it the Alpine strain. This was the variant which arose in the spring, the sort of the half term of 2020, at the time when it's February 2020, at the time when people from all over Europe were going on skiing holidays in Austria and Italy. And there was a new variant which arose at that point and then spread and became the dominant one across the whole of Europe, and perhaps other parts of the world as well, but certainly dominant across Europe by late spring of 2020. And I've called that the Alpine strain. And one of the things that they noticed was there was this change. So the number here, 614, that is the number along that amino acid chain of 1273, and this is the change from one amino acid to another. And so this change was noted, and there was a difference. That was the difference between that one and the original Wuhan strain, and so that's why it was being picked up, and that's how we know from epidemiological studies that it had spread so widely. Then the alpha strain, which was one that arose in, which was first detected in Kent in the end of 2020, had that change, but also some other quite significant ones. So this is a gene deletion at that point, and then amino acid changes from one to another at the point of 501 and 681. Of course, they did have other changes, and this is only in the spike protein. But these are the ones which I think are interesting. Then later on, we found the beta and the gamma variants, and they had some things in common with the alpha variant and also had some other changes. Now, again, those are the things they had in common. Beta and gamma were different variants, and they had some differences in the where they changed and how they changed. But these are the ones which I just want you to note because it's interesting. And then when Delta came along in the spring of 2021, it had some things in common with the original Alpine strain and the alpha variant. It had some things in common with with another part of the alpha variant, but it was a little bit different from the beta and gamma. And then it had some new mutations of its own, which are these ones down here. At the moment, we are dealing with variations on the theme of Omicron, and I've just picked one of the Omicron variants here, it's BA4. And again, what you see here is it has things in common with almost all of the variants along the way. So even including the original Alpine strain, that gene deletion that was important in alpha, and all those other things as we're going along. So it's interesting because it helps us to track what the virus is actually doing, but it also is significant and important to note these things when you're developing diagnostic assays. So the PCR testing and sequencing currently, what we have at the moment in March 2023, is that PCR testing and sequencing of isolates only happening for people who are symptomatic and people who have been admitted to hospital or who are care home residents. That sort of mass community testing is not being done anymore. And we're not testing mass screening of asymptomatic people isn't happening anymore. So the only information we have about what the virus is doing comes from people who are symptomatic and are ill enough to get to hospital. At the time when this presentation was being developed, at the end of February, about 44% of the isolates were this variant of Omicron. But circulating at that time, there are a mixture of at least 5 other variants. And at the time when I'm recording this, this there are other variants which are in the mix. So, and I think this is what's going to happen with the SARS-CoV-2 virus over time, that there are going to be lots of different variants. One or other of them might be dominant at a certain time, but then eventually you'll probably have a mixture of lots of different variants all circulating, particularly in the community. Now, serology was another different way of going down this thing. Particularly, people thought that in 2020, they thought it might be useful for a rapid antibody test. So community testing could be based on testing for IgM or IgG through lateral flow devices. And it was being touted as a potential alternative to mass testing by PCR. But the problem with lateral flow devices in microbiology in general, and particularly the ones that are testing for antibodies, is they have very poor sensitivity. Their specificity tends to be quite good, but their sensitivity tends to be really quite poor. And if you'll remember the timeline that I gave you, what in terms of what happens with the incubation time and the symptoms and antibody being detectable, if you'll remember, it's at least a week before symptoms appear, before IgM becomes detectable, and that is with a very sensitive test. And these were not very sensitive. And so on evaluation, it turned out, as I and probably many other virologists and probably other people other biomedical scientists would have predicted, that it didn't turn out to be particularly useful for diagnosis of recurrent infection. However, it did prove to be quite interesting and useful for mass screening of healthcare work of populations after the event. So, and in this case, the serology, which was used in the main analyzers, was detecting the N protein, the nucleocapsid protein, so antibodies against the nucleocapsid protein. And those turned out to be quite useful because given that at least 1 in 3 people with an active COVID infection who is potentially, it might turn into infection, might turn into illness themselves. They'll also be infectious to other people, don't have any symptoms. So there was, particularly in 2020, it was really unclear how widely the COVID-19 was spreading. So I've just given you an example of a study from May 2020 in Spain, where they had a lot of COVID cases earlier on in the pandemic in Spain and Italy, as well as other countries. And they found that in retrospect, just looking for IgG against the N protein of SARS-CoV-2, they found about 10% of healthcare workers actually had undetectable antibody. The other thing about serology, the other thing which makes it interesting to study, but perhaps not that useful in terms of diagnosis, is it's very unclear how long immunity might last. We know from what we know about coronaviruses in general, common cold coronaviruses, is that antibody protection doesn't tend to last very long. In some cases, there was a study which was published in 2018, I think, of people who were being tested regularly throughout winter season at a big hospital laboratory in New York City, and it was over several winters. And what they did was look to see what viruses people were having. So they were taking throat swabs, seeing what viruses these people had. And there were some people who were in that study who got the same strain of the same common cold coronavirus 3 or 4 times in one winter season, which we knew this in the beginning of the SARS-CoV-2 outbreak, that things, the way that you would expect perhaps protection to come from natural infection against other sorts of viruses wouldn't necessarily apply, definitely wouldn't apply for SARS-CoV-2. But it's been interesting to look at not just the IgM and IgG response, but also T cell response as well. And that research into that is ongoing. Now, the thing that you'll be very familiar with is towards the end of 2020, lateral flow devices became very, very widely offered. And in that case, it was that the people were using a throat nose swab and or a throat swab. And most of the kits which are available across the world, as far as I can work out, detect the nucleocapsid protein. And again, they have very good specificity, but as with other lateral flow devices in microbiology, they have relatively low sensitivity. And so that's why you often find people, that's why people were being encouraged to test themselves regularly, particularly when they didn't have symptoms. And those of you that have had COVID will probably have the experience of feeling ill and thinking, oh, I wonder if it's COVID, doing a lateral flow test and finding that it's negative, and doing another one a day or two later, and then it becoming positive because of its relatively low sensitivity. And that was the rationale for just asking people to test themselves a couple of times a week, and it was very effective method of picking up infection, particularly in people who didn't feel particularly ill. But in terms of as a one-off diagnostic method, it's probably not that useful. One of the things which was unexpected, I think, in a way of this, was that it was being conducted, the rapid tests were being done outside of normal healthcare settings, normally quite often at home. Some of them were at school or at work. We set up the testing centres at the university where I work, but people were also encouraged to test themselves at home. And some bits of it were not particularly intuitively obvious. And so some of that, having worked with my colleagues at my university to train people who are doing the testing, I actually thought that actually the guidance that they were getting could be improved on a little bit. And so we worked with IBMS colleagues to actually provide guidance for people. Particularly, we were asked to help out with people who were doing tests in school. So parents and school teachers are trying to support school children doing tests. We found that there were some things which needed a bit of extra explanation, and so that was something that the IBMS contributed during COVID pandemic. So this, you'll all be very familiar with this, I'm sure. This is the lateral flow test for the COVID antigen, and then you take the throat swab, mix it up with some buffer, put it into this slot here, and then wait for the liquid to move up the cellulose, and you have a thing which detects the control, which detects whether the test has worked or not. There are sort of the colour change chemicals in the test itself will react with the buffer, and then if there's virus present, and the T zone will light up. And this was one I did on myself, and at that time I was negative, which was good. The thing about the rapid tests was they were, as I mentioned earlier, they were designed originally for triage of people who actually had symptoms and then had symptoms for a little while. But they were widely used, and they worked better than expected, I think, on screening for asymptomatic people, mainly because people were testing themselves regularly. And that's, I think, an important thing to remember. If we get into a position again where we're asking people to test themselves at home for anything, particularly an infectious disease, then one-off tests are probably not going to be that useful. But regular testing could play a useful role in infection control and in public health in general. How useful it is in the community does require an understanding when you're setting up the testing regime of positive and negative predictive value and prevalence. And you have to balance. Again, this is something which was talked about particularly outside of pathology labs by the general public. False positive and false negative. There's an awful lot of discussion. People perhaps don't remember it, sort of towards the end of 2020 and into early 2021, people again sort of saying, well, I've tested positive for COVID, but how do I know that it's real? Because what's the false positive rate? And of course, at the time, the risk at the time when we were talking about, so through 2021, for example, there was a much greater risk of it being a false negative result just by statistics and by virological science and by sort of the technical side of the test. But nevertheless, it's worth having those things in mind and having answers for those questions based on science and based on what we know about how tests actually tend to perform. There are other point of care test methods which started being developed more, latterly, and that they include developing molecular methods in order to be sort of benchtop point of care type tests. So there are things for PCR and also LAMP, which some of you may well have been working with yourself, and there's also a CRISPR method which is being developed as well. So obviously they're more expensive and they're not something that you can test yourself at home with, but they can be useful in a clinical setting, and indeed they have been proved to be. So where are we now in 2023? Obviously, people are still getting COVID and coming to require medical attention, coming to hospital, but most of the PCR protocols for respiratory infections are multiplex, and so in most cases, the diagnostic protocol looking at syndromic testing for respiratory infections and the primers for SARS-CoV-2 are incorporated into a multiplex respiratory panel. So it's kind of become more normal and more routine in that sense. But there are still sequencing and surveillance for the variants in the reference laboratories, again, all around the world, not just in the UK, and that's actually very helpful in finding out what's the virus actually up to. And then there are international databases which are collecting all that information so that we can see what's the virus doing in various parts of the world and what are the risks to of one or other of them taking off, and then also sort of evaluating how that works against the vaccine, since people are still being vaccinated. So that's really important information. That's really important to keep collecting that. In the community, again, most parts of the world don't seem to be doing asymptomatic screening, but what we have found is that the rapid lateral flow devices and molecular point of care testing can be useful in the community under certain circumstances. But of course, there's a cost involved. They're pretty expensive. So most governments and most health departments around the world are actually deciding how much they're going to use these things in the long term. But they're always going to be useful if there's an outbreak somewhere of COVID. These things will be useful to implement, because we now, again, as I was saying earlier about the vaccine, we have a lot of information about these tests, so we know how to use them most effectively. So what lessons have we learned so far? One of the things we've learned is that scientists can do things really rapidly. If you give them the time and space and financial resources and laboratory resources to actually do things, they can develop tests, evaluate them, and implement them really very quickly. Also, the other thing which was a little bit surprising, I think, to a lot of us was that mass testing, it's very difficult to take your own throat swab. But mass testing outside of a clinical setting actually can really work. So we were asking people to do something which is really awkward and really horrible, which is take your own throat swab, and then also report your results to the central database. And people did do this, and that turned out to be very useful in terms of public health and infection control. And so that's something that we've learned, that people in the kind of situation that we were in, if you give people the right information and correctly and clearly, then most people will go along with it, and it can be really useful. The other thing that we've learned, I'm sure, from the Lighthouse laboratories is that clinical laboratory professionals have invaluable experience and expertise, and they really should be involved in, if we have to do anything like this again, we really should involve us and our colleagues really should be involved in setting up, and running these laboratories, even if we have non-registered people working in the laboratories. They need to be under the supervision of people who are on professional registers and do know what they're doing. And then also they should have, really proper training. The other thing which really surprised me was we developed a vaccine within 12 months. We had a vaccine. The previous record for developing a vaccine had been for a mumps vaccine, and that had been 4 years between starting work on the vaccine and getting it through all the clinical trials and into routine practice. So that was a major miracle in modern science, was to get the vaccine that quickly. And the other thing, which is even more important, I think, for everybody, is the fact RNA vaccines work. A lot of work had been done on RNA vaccines prior to the pandemic, but they kind of all stalled at certain points. But because a lot of time and effort and money and resources and concentrated effort was put into trying to get the RNA vaccines to work, and they did, we're now seeing the benefits of that is that RNA vaccines for cancer, which was what they were originally being developed for, have actually moved along much further than they might otherwise have done. And some of those are now into clinical trials, and that's really amazing, a really amazing outcome. And I've just given you a couple of references there in case you're interested in following up any of the things which I've talked about.